All right, today we are going to present about Brigalow Woodlands Conservation Project, the application of herbicides and revegetation as methods to control buffalo grass. With me, Arisa Putra, Lewa Tanuki, and also Hui Ting Zhu. I'm going to start with the Brigalow Woodlands background here. Brigalow Belt, it's a really large bioregion in Queensland and New South Wales, Australia. It's a really important ecosystem because of vital for nitrogen fixation, soil fertility, and habitats of many species, native species. The problem is more than 7 million hectares of Brigalow forest have been cleared and converted into buffalo grass pastures for to feed the livestock. And do you know how big it is? 7 million hectares. It's equals almost close to to 10 million of soccer fields. These are really big problems because buffalo grass invasion increases the fuel load and fire risks in the ecosystem, and also it harms the brigalow trees and other native species. Several previous research before. Fire grazing are effective if seed sources remain abundant, so it's not a good option. And also sunny in Arizona in the US, herbicide 3 times or more a year is most effective for areas larger than zero point 0.7 hectares. So native plant cover and richness recovered after herbicide application. Therefore, herbicide may be a solution in Australia to remove buffer grass without losing carbon storage capacity. And this is our research proposal and knowledge gaps here. The first one is limited understanding of the impacts of buffalo grass invasion on native understory plants like bush tomatoes. And also the second one, uncertainty in the best methods to control buffalo without harming carbon storage or conservation values. And what our project will deliver, the first one is testing 2 herbicides and choosing which one is the best one. And the second one is testing revegetation as a method to reduce spread of buffalo grass. And here's our research questions. The first one, how does buffalo grass invasion affect the composition and abundance of native understory plants, including the 2 bush tomatoes? And the second one, how do the 2 herbicides differ in their effectiveness at removing buffalo grass and their impacts on native species of conservation concern?
We divide the area into 200 by 200 meter grills. The yellow area shows our prediction of buffalo grass spread. We marked our study area with yellow stars. Study 1 to 3 will be both conducted in the same grills. To make sampling easier and avoid the effect of herbicides, we slip them into the top and the bottom parts. In study 1, we focus on how buffalo grass affects native understory and the threatened plant species. The first hypothesis is buffalo grass has spread into conservation area, and the second one is the buffalo grass negatively impacts the local vegetation. Our sample size is 10 by 10 meter within each selected grill. We will randomly distribute 5 plots in the upper part. We will collect the data of native understory plants and key threatened plant species. Considering it might be difficult to calculate the buffer grass cover in the whole plot, we randomly locate 10 1 by 1 meter quadrants in each plot. When get the 10 coverage value, we take the average value for the plot. We will collect the data from the following aspects. First, we developed the buffalo grass cover into 3 levels, low, medium, and high, for further analyze. Also, we add the covariate relative light intensity because of the tree difference between the remnant and regrowth. Sampling will be conducted twice a year, in total 6 times for 3 years. In late March to late May, there's a high plant diversity. We can see native species growing and spreading. Also, in September and October, during this time period, plants start to grow, flower, and produce seedling. It helps tracking the invasion and native recovery. We will use a linear mixed effects model to process our data. The first graph is going to support our hypothesis 1. Buffel grass cover increased closer to the edge zone, showing the ongoing invasions. Also, with buffel grass cover increase, the native species abundance and channel index will both decrease. When buffel grass stays at high coverage for a long time, it will cause native vegetation indexes to drop. Also, the high buffel grass cover would bring negative impact on threatened species. Solemn is more abundant in areas with low buffel grass cover. Also, some other threatened species tend to have poor health score when exposed to prolonged high buffel grass coverage. In our next study, we will test the effectiveness of the 2 herbicides for buffel grass removal. We have identified 2 herbicides that are currently being tested for buffel grass removal in Australia and in the US, which are less harmful to trees and plants. We will name them herbicide 1 and 2 in the next steps. Our hypothesis is that the 2 herbicides will have different effects on buffel grass removal, with the trade-off harming native species. Our sampling location will be the same as study 1, but the plot will be distant away to avoid impact of herbicides on our ongoing vegetation survey. In each grid, we will select the 10 plots of 5 by 5 meter to spread herbicide on the top of half and keep the bottom half for control. We will measure the effect of herbicide on buffel grass cover and the effect of herbicide on understory cover by other species.
So as explained, in our methodology, we will have plots where half of the plot is sprayed and the other half of the rest is the control for both of the herbicides. And we will measure our parameters of buffel grass cover and understory cover by other species of interest, returning every 3 months of the same plots. When we return, we will also be applying herbicide multiple times to fully eradicate the buffel grass. The experiment will run from January 2026 to October 2026 to account for seasonal variation. And as mentioned, we will do repeated herbicide applications when we return to measure our parameters. From this experiment, we hope to answer the questions, which herbicides remove the most buffel grass, and also which herbicide is the less harmful to native species. We might expect the results to look something like, as the months go by after application, buffel grass cover will decrease, but at different amounts for the 2 different herbicides. And we want to determine which herbicide removed more buffel grass cover. At the same time, we will also look at the impact on understory cover to see which herbicide may be more damaging to the understory cover that we do want to conserve. By running a linear mixed effects model, we can account for random effects by the placement of the grid, plot, environment type, and season, and really focus on the impact of the treatment of herbicide that might be driving changes in buffel grass and understory cover. So in study 2, we will have been able to determine which herbicide will be more effective for buffel grass removal. And then finally, in our last study, we want to evaluate the effectiveness of combining revegetation to use it as a method to prevent further buffel grass spread. Because as we have known from our research, we need both removal of the buffel grass and then control the spread afterwards. So in this final stage, we will return to the same experimental plots that we had in our study 2. And we hypothesize that the application of whichever of the herbicides is more effective, which we will just say herbicide 1 for now, we will hypothesize that herbicide 1 will not inhibit the establishment of native species, and that the revegetation of native species will outcompete buffel grass and prevent further spread. Here is the methodology. So first, we need to make sure that the buffel grass has been completely eradicated using our chosen herbicide, so we may need to reapply again before starting our final study. And another key preparation for this phase is collecting seeds from native plant species during phases 1 and 2. Also before we begin study 3, we will need to use a nursery to cultivate tube stocks of these species so that they can be used in our revegetation. So finally, once all of these preparation is ready, we will go back to our plots and we will proceed with revegetating half of the sampling plots. And again, we will measure buffel grass cover, which should be 0 or near 0 at this point, and understory cover after we have done our revegetation. We will return every 3 months and measure the same parameters for continuing for 1 year. We will begin the preparation for the study in 2026 and start revegetation in October 2026, which would be during the springtime. And from this experiment, we aim to answer the following. So first, can native understory establish in areas with residual herbicide? Again, we want to be really thorough in our recommendations and make sure that the herbicide we recommend is not preventing revegetation by native understory in the future. And second, does revegetation actually prevent re-invasion by buffel grass? If revegetation can help native plants to outcompete buffel grasses like we predict, we would expect to see buffel grass cover either stay low or decrease over time. This will prevent the further encroachment of buffel grass into the edge zone, and hopefully we can deliver you a two-part solution that will address both removal of the buffel grass and then control of the spread.
All right, let's jump to our recommendations here. The first one, we need to apply the herbicide that produce lower buffel cover with minimal harm to native vegetation. It will impact to the regrowth of native vegetation to restore Brigalow woodlands. And the second one, we need to monitor buffel regrowth rates for at least 12 months to evaluate treatment effectiveness. It will to reduce fuel load leading to reduced climate risk. And the last one, we need to identify collaboration opportunities with other local Brigalow conservation groups and also local people to share evidence-based best practices. And we believe that it will increase long-term capacity for carbon storage. Thank you.